Good morning, everybody. Give everybody a couple of minutes. Yeah, no, what a wonderful break, right? You know, kind of, it's like being in a cabin in the woods or something like that, I guess. No electricity, cuddle up with your family, stuff like that. Lots of fun. Irony is that I've lived in places with far, far worse winters than what we experienced and never experienced a blackout. It's only in Texas, which is I find kind of ironic. Yeah, I guess I mean, it was kind of a bit of a chore having to reschedule everything, but I didn't have to deal with homework and the like. So hopefully everything's kind of back to normal for everybody by and large. So for those of you that are here, uh, everybody in my lab section has already received an updated schedule. You should be receiving an updated schedule from Mr. Penny soon if you haven't already. If you haven't, please do feel free to email him and ask him for it so that you can kind of get back on track for the lab. Does anybody have any questions about anything before we get started? I think I've ticked all the boxes of stuff that needs rescheduling and rearranging and things and sent out announcements about them. But if I haven't, please do let me know and ask. Oh, right. So no one's got any questions? Feel free to chime in whenever you do, but if you don't, we can get started. So, doopy-doo. And we've got chat. Oh, thing with the lab is the lab schedule has changed. So that it's basically everything's shifted back a week, but you'll still be attending on the same weeks that you were originally, like the alternating week thing. And basically, it's all been pushed back a week so that open lab at the end of the semester no longer exists. And I've sent out an updated schedule to my lab section. Mr. Penny has the same schedule. He should be sending it out too. If he hasn't, please remind him. He should have done that already. So yeah, all the details will be in the new schedule. Did that help, JM? It's kind of hard for me to keep track with who of you are in my lab section and who aren't, to be honest. It's all a little bit crazy. Okay, so essentially what we're going to be covering today. So the next, was it 3 lectures, I think, is all going to be about various exceptions, extensions, whatever to Mendelian genetics, right? So Mendelian genetics, you know already, the law of dominance, law of segregation, law of independent assortment, right? Basically rules by which we can figure out the likelihood of inheritance of different traits, right? Probability, basically. So obviously all those laws are very much about ideal scenarios. We already talked a little bit about the assumptions that are kind of implicit in those laws. And obviously real life, real biology is not that straightforward and simple. So there are lots of ways in which those laws are broken, for want of a better word, or circumvented, right? You know, that they don't always apply the same way. And so really what we're going to be doing over the next 3 lectures is looking at some of the ways in which those laws don't quite work, right? The way that they should in theory. And so today we're going to be talking about sex determination and sex-linked inheritance. Actually, I wonder if we can British. Come on. Get this up while I remember. There you go, that's as good as any. Oh, tiny ass picture. Yeah, okay, well, we'll go back to that in a second. So first we're going to be talking about sex determination and sex-linked inheritance, which are different things, but they're all about the sex chromosome. Then we're going to be talking about essentially different types of dominance, which you're probably somewhat familiar with already, codominance and incomplete dominance. And then we're also going to be talking about epistasis, right? How one trait can mask the phenotype of another trait, essentially, which completely balks up the law of independent assortment. Well, kind of, yeah, sort of, yeah, maybe. And also we've got a absolutely huge section, where are we, on linkage, right? So this is something that really messes up the law of independent assortment, right? So this is a big stretch of stuff all about what happens when 2 genes are close together on the same chromosome, right? Because our assumption for law of independent assortment is that they are on different chromosomes, right? And obviously we have 23 chromosomes and about 20,000 genes. So there are going to be a lot of different genes on each chromosome. Okay, so that's essentially kind of the plan for this section, right? It's kind of like all the ways in which Mendelian genetics kind of doesn't work, right? In a sense, right? And that's actually the fun stuff, right? It's not, I mean, monohybrid and dihybrid crosses are all well and good, but they're not particularly interesting. Things like this are super fun to talk about. So Russian royal family, there's Tsar Nicholas and his wife, that one there. All of them murdered, unfortunately, by what is it, the Red Russians? They were called at the time, communists as we now know them as, after the 1917 revolution. Found buried in a shallow grave in Siberia or something like that. So the fact that Alexei had haemophilia was a bit of a moot point, to be honest, but is kind of a classic example of that. Anyway, a little bit of history for you there. He wasn't very well liked by a lot of people in Russia, hence his demise. So anyway, I'm not going to talk about them too much. It's just a nice picture, bit of history. What we're going to talk about first is sex determination, which is a pretty funky topic, right? And so really this is about how dioecious species, dioecious means sexually reproducing, so typically male and females, how dioecious species determine their sexual phenotype. And there are a lot of different ways in which they can do that. Some, the environmental ones are always fun to talk about because you're probably familiar with the whole alligator eggs in sand, kind of what temperature that is determines the sex, right? And so lots of different reptiles rely on temperature for sex determination. Don't know how they do that. I hope it is known, but I don't know how it's done. Some actually, this is like a real funky kind of topic to talk about, because this is like, oh yeah, that kind of makes sense, right? For some weird reason, right? They use temperature. Obviously not in Texas, right? That would have been disastrous recently. But fish are even weirder. So fish, some fish, not all fish, yeah, even weirder than that generally. Some fish change sex depending on how many members of that sex are around them. So this isn't like my offspring changes sex depending on environment. This is me. Yeah, crazy. So if there aren't very many males around, some of the females will change into males, right? To kind of even up the sex ratio, which is really, really weird. I don't understand that one little bit. But anyway, so environmentally, obviously, is one way in which you can control your sex if you're the right type of species. We're going to go into kind of some chromosomal and molecular mechanisms in a bit, but the one bit of terminology that's very important is the difference between sex chromosomes and autosomes, right? So autosomes are all the chromosomes that don't differ between sexes, essentially, right? It's probably the best way. I mean, some autosomes do have a role in sex determination, but they are the same whether you're male or female. It's kind of figured out a different way, right? Drosophila are a good example of that. Whereas sex chromosomes differ between either in number or size, shape between males and females, right? So this is a very important piece of terminology to remember. We'll be using it a bunch. I think, sorry, I've just got to go let my puppy out because I don't want him to pee on the floor. Won't be a second. Here we go. All right. Hopefully he's going to be doing his business quite happily out on the lawn. We got one of our foster puppies back because he hasn't been adopted yet. So we're having to figure out how to get him to not pee on the floor, which is kind of interesting. Anyway, so there are lots of different ways that this can be done, right? Pardon me. How we determine sex based on chromosome, how many of those different sex chromosomes you have, right? We're obviously all very well familiar with this whole XXXY deal because that's what we have ourselves, right? So females are homogametic, right? They have two X's or two chromosomes that are the same, sex chromosomes. Males are XY, right? So we have one X chromosome and one small, diminutive, tiny, stunted Y chromosome. That's not the only way, right? So in mammals and some insects, Drosophila, for example, it's XXXY, where the heterogametic is male. Birds, like chickens, it's the other way around, right? So they have the same X or ZZZW, but in this case, the ZZ chickens are roosters. They're males, whereas the heterogametic are female, right? Not very familiar with how this all works in birds, to be honest. It's not something that I've spent a whole ton of time looking into. But there's another way, which is also, this is called, the term is chromosomal diminution, right? Which basically means one of the X chromosomes disappears. So some insects, definitely nematodes, right? So little tiny roundworms use this. One of the things I worked on. Actually, even C. elegans, what I work on now, uses this mechanism for sex determination. So here, XX are female, right? And in the process of generating sperm, one of those X chromosomes is lost, right? So sperm, the male gamete, doesn't have an X chromosome, right? Just doesn't have one at all, right? There's no other type of chromosome. It just is a lack of X chromosome, right? So in this system, it's a matter of how many X chromosomes you have, 2 or 1, determines your sex. Actually, that kind of works on Drosophila too. This gets all kinds of crazy, trust me. This is something that I've spent a bunch of time reading up on so that I understand it well. One of the funky things too is that both mammals and fruit flies have the same XXXY sex determination, but it is very, very different how that works, right? So I'm going to actually draw this up because this is a little bit confusing, and it will help me to kind of put my thoughts straight to explain it to you all. Right, so basically in Drosophila, it's all depends on this particular gene and protein. This is stands for sex lethal, right? And it is an RNA binding protein, right? Probably one of the more famous ones. So I'm going to, does it make a difference if I stop the share and how big you see me? I know it does in WebEx. I don't know about in Zoom. Anybody? Does my picture get bigger if I stop sharing?
Yes, it does.
Oh, okay, cool. Thank you, whoever that was. Okay. Let's see. Are we going to do okay with reflection and the like? Okay, so pens. Basically, in Drosophila, there you go. There's a Drosophila. Right. What they do is they actually count, obviously not, with their fingers or whatever they have, but they count how many X chromosomes they have to autosomes. So the deal is the chromosome to autosome ratio. Right, with humans, it's just, well, do you have XX or do you have XY, right? And we'll get into that in a little bit. With Drosophila, it's how many of these do you have to these, right? So this is fixed. How many autosomes you have as male or a female stays the same, right? Because they don't differ between the sexes. This is what varies, right? So Drosophila and they have 3 chromosomes. I think they have 4, but one of them doesn't really do very much, I think. So what we have in females is we have XX. So we have 2 sex chromosomes. We have a fixed number over here of 2, right? In males, we have XY, and so we only have 1 X chromosome. So females are actually 2 to 2 sex ratio, sex chromosome, sorry, to autosome. Males, this is kind of a pain. I wonder if I just take this out. Sorry, rearranging the couch. Hey, found a pen. It's always useful. There we go. Look at that. That's even better. Sort of. There we go. Now we're cooking with gas. All right. Isn't that neat? Whereas males are one X chromosome to 2 autosomes. Okay, everybody follow along so far? I'm just going to take a picture of that while you kind of let stuff sink in.
So the autosomes are always 2. That's just a constant that we can rely on.
Correct. Yes, it's very much the same way that we always have, what would it be, 44 autosomes, because our total chromosome count is 46. So humans, whatever sex your cells are, or you are, you always have 44 autosomes. And it's the same in flies, they always have 2. And so that
So the reason
go on.
The reason for the huge difference in autosome, is that just because we're much more complex organisms than they are?
Yeah, in many ways. That's actually a really interesting kind of bigger picture question. Kind of evolutionary history, I think, is one kind of easy answer, right? So we've accumulated a whole bunch of other junk which fruit flies don't have, because most of our genome is non-coding, right? It doesn't encode proteins, vast majority of it actually. Yeah, I mean, it's actually, it's kind of an interesting point. So fruit flies have approximately the same number of genes that we do, within a few thousand at least. But we have far more chromosomes than fruit flies do. So as a general rule, the more complex the organism, the higher the number of chromosomes you have. But that doesn't apply to the number of genes that you have, or actually the amount of DNA you have either. It's a very weird sort of phenomenon to talk about, right? So much of the complexity that we have as humans over flies, fruit flies, is it's actually down to how we control the expression of those genes, right? We have many, many, many more ways of controlling those, and in some ways we have more variants of the same thing, right? But it's not down to how much DNA we have, and it's not down to how many genes we have. It's just, yeah, I know. It's kind of it's a very difficult question to answer, to be honest. But generally speaking, chromosome count scales with complexity, generally speaking, but not necessarily for the reasons that you think it does. Sorry, that probably didn't help very much, Amber, but it's it's one of those sort of cop-out questions, unfortunately. Cop-out answers.
No, it's
rather.
okay. I'm just trying to find a way to understand it. I like to compare it to us to kind of get the
Yeah, so
the understanding.
yeah, and it's a very, very interesting question, because superficially, I mean, humans have the same number of genes as soil nematodes, right? Soil nematodes have 959 cells. We have, I actually don't even know how many, billions probably, right? And we're clearly far more complex than worms are, but we have the same number of genes as they do. So there's something else which controls that or contributes that difference in complexity. I got a question from Rogelio. That's an interesting question. So two different things with that. One is that the male fly has the same number of sex chromosomes. It has 2 sex chromosomes, just as the female fly has 2 sex chromosomes. However, when it comes to, and we'll get to why in a little bit, when it comes to why males are males and females are females, only the X chromosomes count, right? So that's why we have 2 to 2 for females, because we're counting the X chromosomes, we're not counting the sex chromosomes, whereas males are 1 to 2. So they still have the same number overall of sex chromosomes, but they only have 1 X, right? And we'll see why that matters in a little bit. Now, there's also something else which is pretty funky when it comes to this difference in number of X's, right? So essentially in humans, females have twice the gene count for the X chromosome than males do. So in humans, human females, one of those X's is inactivated. In every single one of all the ladies that are present, in every single one of your cells, one of your X chromosomes does not work, right? It's shut down, heterochromatin, so that we have an equal number of functional X chromosomes. Like males have 1, females have 1, and it just varies which cell, which chromosome is inactivated in which cell, right? That's actually where sex linkage comes in and sex-linked diseases, but we'll get to that later. In fruit flies, it's the opposite. So in fruit flies, they also have XXXY, but rather than shutting down one of the X's, they double the amount of expression from the 1 X that they do have, right? So overall, you have the same amount of, say, gene X1, right? Produce a normal level from both of these chromosomes in females. X1, you've only got 1 copy of it. In males, and it's produced at twice the level, right? So it's called dosage compensation. How you deal with this difference in X chromosomes in terms of gene expression differs between species as well. So the number stays the same, but the functional consequence is different because of the number of X's, and the X's in Drosophila are what count. The Y does not. In humans, it's different. The Y is actually pretty important in sex determination. Any other questions? But the one thing to remember is that this 2 to 2 ratio female, 1 to 2 ratio male. And you can get some real funky kind of chromosome counts in like weird flies, which we're not going to talk about very much because I don't really understand it. But you can get more than this number of chromosomes and still get a functional fly. Okay, so I'm just going to put back up here as a reminder. Sexed autosome, sexed autosome, right? And so how do they use this kind of chromosome counting, right? And so what happens here is we have autosomes repress or something produced from the autosomes. I'd have to think really hard about what that is because I've read about it. I can't remember off the top of my head. This particular protein, which is produced from X chromosome. And this is one of those funny examples where I'm kind of teaching you how I learned it, because every single time I teach this, I have to go over and teach myself again how this works. And every time I get a little bit better at it. And I think I've fully got it now after like the third or fourth time. Right. So this is kind of how I rationalize how this works. Right. So we have 2 times repressor. Right. Well, let's put 2 lots so we don't confuse X's and times, right? Right. Because we have our 2 autosomes. Now in females, we have 2 lots of sex lethal because it's produced from the X chromosome. Now in, and also I did tell you about the dosage compensation, but that doesn't quite play in here, right? So I don't kind of confuse the issue. Males have 1 lot of sex lethal. So basically what we have is sex lethal promotes female traits and inhibits, that's what that kind of blunt arrow is, male traits. Everybody following so far? Stop me if you're not, so I can go back through anything. Okay. Right, so again, the key point here is that sex lethal pro-female, anti-male. It's not personal, it's just that's the way it works, right? And females have 2 lots of sex lethal, 2 doses, let's say. Males only have 1, right? This amount of repressor stays the same. So essentially, in males, we have 2 lots of repressor turns off 1 sex lethal equals sex lethal off. So if sex lethal is off, and so this is, if we have a 1:2 ratio, right, which is male, sex lethal gets turned off. We no longer promote, sorry, making sure I get, let's tip this down a little bit. There we go, that's better. Sex lethal gets turned off because there's more repressor than there is sex lethal, right? Because we only have 1 lot of sex lethal. If sex lethal's turned off, there's a whole bunch of stuff which kind of follows on from that. But basically, we don't have female traits anymore because sex lethal promotes femaleness. We get rid of this inhibition of male traits and we end up with a male. Right, I'm just going to take a picture of that and I'll go through the same on, come on. Make sure I get. Now, get rid of all this. Back here again. Now, if we have a 2:2 ratio, which is going to be female, now we have the same amount of, well, we have more sex lethal, right? And so sex lethal is not turned off, right? So now it inhibits male traits and promotes female traits, and now you have a female. Dang it. This is, there you go. That's better. I'm getting some, it's from my bloody laptop thing. So depending on how much sex lethal you have, which is determined by how many X chromosomes you have, determines whether or not that sex lethal is going to get turned on or off, or is just going to remain on or get turned off. If it remains on, because there's essentially less repressor per unit of sex lethal. If it stays on, then it inhibits male. Oh, hopefully my internet isn't going to crap out. It inhibits the male traits and promotes female traits. If there's less sex lethal, because we only have one lot of it, because we're males, sex lethal is then repressed. That means that female traits don't work, which means we have males. Ta-da! Told you it was crazy. Let me take another picture while I'm waiting. Anybody have questions? Oh, okay. So you can actually have fruit flies that are XXY. These are the weird ones that I was telling you about, right? And so if you have an XXY, you'll have, I think they call them super females, right? So you'll have, they will still be female because the Y doesn't contribute, right? The Y doesn't really do anything. And so you'll still have 2 doses of sex lethal, and sex lethal will be on still. And so it will promote the female traits and you won't have males. That's in fruit flies. If you have that in humans, that's actually called, Klinefelter's syndrome, where essentially you have, I wouldn't call it intersex, but you have both sexual traits being expressed, but the general outcome is that the individual is sterile. They won't have functional reproductive organs. And the same happens, you can have, what is it, just single X, I think. Oh yeah, Superwoman. Yeah, I don't know about that. You look up Klinefelter's. It's really curious to look at the actual physiology of the individuals that have that. I think you can have just X alone, so X0, which is also still functional, but also I can't remember what syndrome that's called. That's also that individual will also be sterile too. But yeah, anyway, it's kind of crazy. Anybody got any questions about fruit fly stuff before we move on? Cool. Okay. So in humans, right, we have exactly the same. Yeah, all complicated. That's why genetics is fun, right? None of it's simple. This is why I find, let me just rub this off before I forget. This is why I find the non-Mendelian genetics part so much more interesting than Mendelian genetics, because Mendelian genetics is just like, you know, whatevs, kind of, it just happens. It's pretty straightforward. It's all logical. It's, you know, I wouldn't say simple, but it's not super complicated either. It's when you get into the weeds of non-Mendelian genetics that stuff gets really funky, right? So in humans, it's different, right? So in humans, the Y chromosome does matter. And it matters because essentially it doesn't contain many genes. It's very, 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 very small. And one of the main key regions, it produces two things, right, which kind of really matter, right? One is production of testosterone. Two is what's called SRY, or the sex determinant region Y, SRY, right? And basically what happens is that early on in development, up to, I think it's like 7 weeks post fertilization, your gonads are undifferentiated. They're actually called indifferent, which I think is kind of funny. They're indifferent. It's like, I don't care, like a teenager, I guess. But they do not have a sex, right? And so at that point, they have two sets of what are called ducts. It's like, kind of hot air ducts that you have in your house. They have what are called Mullerian ducts, and they have what are called Wolffian ducts. Let me write that out because that's kind of fun. Yeah, I love the fact they're called indifferent. And Mullerian are what are called proto-female, right? And I think it's Wolffian. Maybe got the spelling wrong. Right. And essentially, at that point, they can become either male or female. So if that embryo has XY genotype, essentially what happens is that region on the Y chromosome produces a transcription factor that turns on kind of pro-male development, right? And it does so by promoting the development of the Wolffian ducts, which will then develop into the testes, right, and the epididymis, and all the other kind of stuff that goes with that, right? And the testes also with actually no, the testosterone can't be on the Y chromosome because females produce it as well. There's something else that's also important on the Y. I can't remember right now. But anyway, testes produce testosterone. And testosterone it's not a male hormone, it's a hormone that produces lots of developmental changes. But of that are some key sexual characteristics, right? Also, and this is actually probably more important in terms of sexual differentiation, the testes also produce something called the Mullerian inhibiting factor, right? And what that does is cause the proto-female ducts to degenerate. And so you have like this kind of two-pronged approach, right? We have pro-male, testicles, produce testosterone, stuff like that. And we also have an anti-female kind of prong as well, which is telling those proto-female things, ducts, to disappear, right? And that's how you end up with a male. If you are a female, obviously you're XX, you don't have those signals, right? And instead, what you have is production of estrogen, right? And estrogen promotes the development of the Mullerian ducts, which then develop into ovaries and uterus and things like that, and also inhibits the development of the Wolffian ducts, which then just get absorbed over time. There's also funky stuff around there with kidneys as well, but I can't quite remember that off the top of my head, right? So here we have, because in fruit flies, it's very different, right? It's determined like for each individual cell, essentially what sex they are, right? And it's done from the very moment of fertilization, right? In humans, it takes a lot longer because we have these kind of more whole organism processes that kick in, right? Instead of saying at individual cellular level, now what we're doing is saying this particular organ develop in this particular way, and then that will start producing a signal which will tell other stuff to develop in a particular way. That's how we have that kind of split between the males and females. So up until 7 weeks, you are neither, right? You're genetically male or female, but physiologically there, you're not one or the other, which I think is pretty cool. Any questions about that? No, other dog coming down the stairs. Okay, so just to finish up, we're not going to get through this today because we only got 7 minutes left. But I want to get started on sex linkage, right? And so sex linkage is basically, let's see if I can get, is this going to be a good image? Oh yeah, fabulous. Haha, Royal Britannia. This is the British royal family. A whole bunch of inbreds. Not terribly so. I mean, they're better than the Spanish royal family, but still. Okay, essentially what we're looking at here, this is a family tree, right? And what this is showing you is that we have a disease which you only, and this is the red circles, you only see that disease in males, right? This is like the defining characteristics of a sex-linked disease or sex-linked inheritance, right? You only see that phenotype in males, right? And so if you look at this lineage, right, here's Queen Victoria, dour old lady that she was. Presumably before that point, let me get rid of that. She wasn't so dour in that she could have, what was it, 4 kids? I wish I had more than that. Anyway, but Queen Victoria was a carrier, right? So for sex-linked diseases, and the classic ones are hemophilia, which is what we're looking at here, which basically is that you don't produce a clotting factor. And so if you get cut, then you can bleed to death, right? Even from a minor cut. Male pattern baldness, right? So why guys go bald, basically, is also X-linked. And color blindness is another one, right? These are all classic X-linked diseases, right? And because females have 2 X's, right? Two X chromosomes, it's very rare for them to be homozygous recessive for this trait, right? Typically, they are, if they're going to have a recessive allele, they'll be heterozygous, right? So they're called carriers. There's the light pink here. Pardon me. And so in X-linked inheritance, you'll have female characters is Alice, Beatrice, another Alice. That wasn't very imaginative. Another Alice, Victoria. They're not very good on like new names in royal families, it seems. Anyway, all these pink individuals are females and carriers. Now, if you have a carrier, you possess one allele of that disease, then there's a chance that you're going to pass that on to your male or female offspring, right? So if you look here, Victoria, the second, I guess, got an allele. Alphonse, I don't think he was in an American sitcom show. He did not. So he does not have the disease. But both Moritz and Leopold von Battenberg got a X chromosome with a recessive allele, right? And so, long story short, if you have only one X chromosome and you have a recessive allele, you're functionally the same as being homozygous recessive. You're not homozygous recessive because you only have one chromosome, right? That has that gene. But, hey, Soph, just a little quiet, sweetheart. I'm still teaching, okay? But you don't have another copy of that gene, potentially a dominant copy, dominant allele, to cover up that recessive allele. It's essentially what you have in female carriers, right? And so if you only have one chromosome and that chromosome has a recessive allele, then you will express that trait. So any male that inherits that allele, right? So you get it coming down through here. This is actually fairly rare that individuals lived with hemophilia, males with hemophilia lived long enough to reproduce. But Leopold clearly passed his mutated X chromosome on to Alice, not to Alexander, because that wouldn't work. He would have the Y chromosome, but pass on to Alice, and then Alice pass on to Ruprecht, right? And so all of these males have a single X chromosome, obviously, and they have a recessive allele for hemophilia. Essentially, it's I think it's a thrombin, a non-functional thrombin gene, right? And generally speaking, most of these individuals, these male individuals, died before the age of around 20 or so, simply because it's all in that era it was almost impossible to avoid minor injuries, and we didn't have the kind of technology that we have now to prevent runaway bleeding, basically. So anyway, so we're going to look at next class. We're going to finish this off, dunk, and look at some more funky stuff about the Y chromosome and sex linkage, and also how you can actually use that to follow lineages down through, thousands of years. That's pretty much it from me. I'm going to, if you have any questions, Alexander, ooh, I don't think so. I'm a little bit leery of saying that for definite generally, because European royalty is like, interrelated up the wazoo. So I don't know. I don't think so, but I'll look into it because I think Tsar Alexander came from, yeah, probably, from the German lineage side of things. And I know that Queen Victoria came from the Habsburg lineage. Sorry, barking puppy. Which was also from Germany, right? So we kind of imported our royal family when the existing royal family kind of putted out, I guess. So maybe I'll look it up because that would be interesting to find out for sure. Cool. All right. Thanks very much, everybody. I'm going to call it a day for today, and I'll see you on Wednesday. All right. Take care, everybody.